Regulation of gene expression, Chapter 18. We're going to look at regulation of gene expression in both prokaryotes and eukaryotes. What it means is that sometimes you turn genes on and sometimes you don't. So for a eukaryote, there might be times where you turn on genes in neurons and you don't turn them on in skin cells. For prokaryotes, it's just one cell, and so sometimes they're going to turn genes on when they need them, and sometimes they're not. So what I want to look at first is why this would happen, and then we'll look at how it happens. So why this would happen is natural selection. Natural selection has favored bacteria that can regulate gene expression. Why is that? So we're going to look at an example of bacteria that live in your intestines. Some bacteria, the bacteria that are living there are going to be the ones that are most fit because their ancestors survived and reproduced. So let's take an example of bacteria that live in your intestines and what happens when you drink milk, or what happens when you don't drink milk. So milk is a food that has a sugar in it called lactose. So remember, os means sugar. So if you have lactose around in your a bacterium and there's lactose in your environment, you should break it down if it's around, unless you have something better to eat. So when your human has eaten lactose, you should make the enzymes to break it down. And when your human hasn't had any milk and there's no lactose around, then you shouldn't make the enzymes. So let's look at some bacteria and decide which ones are the most fit. So here's contestant number 1, the first bacterium. This one never makes the enzymes to break down lactose. So that one's not going to be super fit if your human is drinking lactose pretty regularly. Here's contestant number 2. This one always makes all of the enzymes to break down lactose. It takes a lot of energy, but contestant number 2 is going to be more fit than contestant number 1 if the human drinks lactose a lot. Here's contestant number 3. I make some of the enzymes to break down lactose sometimes, but I don't make them at the same time very much. So contestant number 3 is just lousy because if you're going to have one enzyme to break down lactose, that's not very useful. You really need all of them to be expressed at the same time. So contestant number 3 is lousy. Here's contestant number 4, who's pretty fantastic. I make all of the enzymes to break down lactose, but only when lactose is around. When lactose isn't around, I don't make the enzymes to break it down. So contestant number 4 is incredibly well fit. So this one just makes the lactose when you need it. There's one more that does even a better job, though. Last one, contestant number 5. I hate to brag. Yeah, you do. But anyway, like contestant number 4, I make all of the enzymes to break down lactose, but only when lactose is around, right? So you're making the lactose here. When lactose isn't around, I don't make the enzymes to break it down. So it makes the enzymes when it needs it, but not when it doesn't need it. That's really, really good. On top of that, when glucose is around, I don't make the enzymes to break it down, even if lactose is too. So here's the thing, glucose is actually an easier food source to use. So this one is going to have such nice tight control over gene regulation that even if lactose is around, it's not going to make the enzymes to break it down if there's something easier to use. So we're going to look at how contestant number 5 has evolved and what systems contestant number 5 actually uses to do this incredible work. So we're going to look first at just what contestant number 4 does. It's called negative gene regulation. And then we'll talk about this extra step here, which is called positive gene regulation. So if bacteria can tightly regulate gene expression, they're going to be more fit. More fit means they can survive and reproduce. Chapter 18.1, bacteria often respond to environmental change by regulating gene expression, or gene transcription. So natural selection has favored bacteria that only produce the products they need when they need them. So here's an example of feedback inhibition, which you've seen before a million times in this class. So this is really not the topic of today's lesson, but I want to compare it to what we're doing today. So we've seen before, if you have a precursor right here, you need an enzyme to modify it and make it intermediate number 1. And then maybe another enzyme to modify that and make intermediate number 2. And then another enzyme to modify that to make what you're finally producing, the product. In this case, the product is tryptophan. Tryptophan is just an amino acid. That amino acid might be around in your intestines, in which case your bacteria can use it. If it's not around, your bacteria might need to make it themselves. So this is called a cascade of reactions. And here's tryptophan. If you have a lot of tryptophan around, it makes sense that a lot of tryptophan around would come back here and turn off this enzyme. So that way you don't keep making more and more and more tryptophan. If tryptophan's not around, then you'll stop breaking down, then you'll stop inactivating the enzyme and you'll make more tryptophan. So that's called negative feedback or feedback inhibition, and we've looked at that before. What we're doing today is a little bit different. What we're looking at today is actually making the enzymes in the first place. So where is my cursor? Okay, so here's enzyme number 1. If you have lots and lots and lots of tryptophan, not only will it turn off enzyme number 1 with this, what we've seen before, this negative feedback, but it's also actually going to turn off the DNA, right? This DNA from making the tryptophan. So lots of tryptophan will come here and it will turn off the genes that you need to make the enzymes. So not only are you turning off the enzymes, you're not even making the enzymes in the first place. So that's called gene regulation, and we're going to be looking at that today. The way that bacteria express genes, or the way that bacteria control gene expression, is with the operon. And so this is different with eukaryotes. We do not use the operon model. Okay, so here's a whole picture of the operon model, and I'm going to be looking at each piece of it with you, but let's just kind of run through the whole thing. So we're going to be looking at the trip operon, which is short for tryptophan. So these are polypeptides, and these are the enzymes that you need in order to make tryptophan. So back to here, these enzymes right here, we're going to be looking at the genes to make those enzymes. Okay, so the DNA, that's this part, codes for RNA, which is this part. And notice how these genes, TRPE, TRPD, TRPC, TRPB, TRPA, those are all transcribed in one whole entire string, which is kind of unusual. That's not what we've looked at so far. So here's the RNA, and notice that the RNA is all together in one big long string. There's a start codon and a stop codon, and so the ribosomes are going to start at this start codon and end at this stop codon, and they'll make this first protein. And then they'll start here at that start, and then this stop, and they'll make this next polypeptide, and then they'll start here and here. So you have a whole series of start and stop codons, and they'll make this one. So the RNA is continuous, but the proteins will be made, the polypeptides will be made in individual units. So it's kind of a cool system because the genes to make these enzymes are all transcribed together or not transcribed at all. So it makes no sense to make this enzyme, but to not make any of these enzymes, right? They're all turned on and off together. So here's the promoter for all of them. And the promoter, remember, is the piece of DNA that RNA polymerase binds to. If RNA polymerase can bind to the promoter and bind pretty well, then you'll transcribe all of these genes, or if the RNA polymerase can't bind, it's not going to transcribe any of these genes. The promoter has a part in it called the operator right here. The operator is really an on-off switch. So I'll run through all of this together in more detail, but if the operator is not blocked, RNA polymerase can bind, and you'll make all of these you'll transcribe all of these genes, make all of these enzymes, and you'll be able to make tryptophan. If something stops the operator from working, or blocks the operator, let's do it that way. If something can block this operator, now RNA polymerase can't move on and you won't make any of the genes. So let's look at each of these pieces. So the operon is defined as the entire stretch of DNA, which includes all of the genes for the enzymes, and it includes the promoter, and the promoter part of the promoter is the operator. So the operator is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. The genes are functionally related. So functionally related means they're all for the same thing. In this case, they all produce tryptophan. In another case, they might produce leucine. It might be the leu operator. These ones making stuff, these are anabolic operons. There are also other operons that break stuff down, like maybe breaking down lactose or breaking down sucrose. These are sugars that a bacterium might use for food. And so those are catabolic operons. So we're going to start talking about anabolic operons like the trip operon. And then later on, at the end of this, we'll talk about catabolic operons that break down things like lactose or sucrose. So these genes right here, they're functionally related, meaning they're needed to work together in order to do something, in this case to produce tryptophan, which is just one random amino acid. Okay, so the operon genes are coordinated by a single on-off switch. The on-off switch is this, the operator. If that operator is blocked, you can't do any transcription of these genes. If the operon is open, if the operator is open, meaning not blocked, then you can go ahead and transcribe all of these genes and make your proteins. So just to define that again, operator, it's usually positioned inside the promoter. Usually we define it as inside. I mean, you could do it either way. But anyway, this is a segment of DNA that acts as an on-off switch. The way it works, see this protein here? This protein right now can't bind in there, but if it could, if it were bound right in here, then it would block the RNA polymerase from binding and going ahead and transcribing. So the operon can be switched on and off by a repressor. The repressor is a protein, and the repressor prevents the gene transcription by binding to the operator and blocking RNA polymerase. So here's a protein that can actually bind to DNA. How cool is that? So this protein is the repressor, and if it's bonded to the operator, that's this, then RNA polymerase can't bind onto the promoter, and the whole transcription can't take place. So that blocks RNA polymerase. So you need to somehow have a repressor around in order to turn the operon on and off. And so that repressor is made by this regulatory gene. So it's, in this case, upstream, but it can be anywhere. Upstream means just earlier in the DNA. So here's the repressor. It's a protein. The repressor is a protein, so that means it's made from RNA, and that RNA came from DNA, and that's right here. This is the regulatory gene. The regulatory gene itself also has a promoter, but it doesn't have an on-off switch. So this is always made. This regulatory gene is always on at a low level, so you've always got some of this repressor around. This repressor is inactive. Look at the shape of it. See how this shape doesn't look like it could actually fit onto the operon? So this is an inactive shape. If the co-repressor, in this case it's tryptophan itself, if the co-repressor attaches, see how now the shape of the repressor is different? That repressor can now bind right on here onto the operator. So you actually need the tryptophan in order to bind to the repressor to make the repressor active. If the repressor is active, it binds to the operator, and now you can't do any transcription of this operon, which makes sense. Because if you have plenty of tryptophan around, you don't want to turn on the enzymes to make more tryptophan. So we're going to call it negative gene control anytime you have a repressor. So whether that repressor is made in an active form or made in an inactive form, if you are talking about a repressor, we're going to call that negative gene control. In anabolic operons like this one, where you're making something, usually the repressor is made in an inactive form, meaning you're just always transcribing these genes, you're always making these enzymes, and you're always producing your product. You're always making tryptophan. But if you have a lot of tryptophan around, it's actually going to make this inactive repressor active, and it'll bond on here to the operator. So the repressor can be in an active or inactive form depending on the presence of other molecules. In this case, this anabolic operon, the co-repressor is tryptophan. So you need this co-repressor to bind to this repressor, and if they're bonded, then we get an active repressor. The active repressor bonds on here to the operator. That stops RNA polymerase from binding, and now you don't make any of the enzymes that you need in order to make tryptophan, which is super duper because you already have plenty of tryptophan around. So remember, this is a little bit different from negative feedback. With negative feedback, you have the tryptophan, and it takes the enzyme and makes it inactive. With the operon system, what you're doing is actually not making the enzyme in the first place. Okay, so by default, the operon and the operon is by default on, meaning the repressor, where am I? The repressor here is inactive, which means you're not stopping RNA polymerase from binding, which means you're going ahead and transcribing all these genes. So by default, the operon is on. When tryptophan is present, here's tryptophan. When tryptophan is present, you are actually activating the repressor. The repressor is binding to the operator. When the operator is blocked, now the RNA polymerase can't bind and you can't transcribe these genes into RNA, and then there's no RNA around to translate it into these proteins. So now you're not going to make tryptophan, which again is super duper because you have plenty of tryptophan. So the default way that this works is that the repressor is usually inactive, the genes are usually transcribed. If you have plenty of tryptophan around, maybe your human ate turkey and you have a lot of tryptophan, now that tryptophan acts as a co-repressor, turning off the whole thing. So the repressor is active only in the presence of the co-repressor tryptophan, so the operon is turned off or repressed only if tryptophan levels are high. There are two different types of operons that we're going to talk about. There are repressible and inducible operons. The repressible operon is the example of tryptophan that we just talked about. The inducible operon is going to be the next one that we talk about. Both of these cases are actually still going to be called negative gene regulation because both of them are going to have a repressor, whether that repressor is normally turned on or normally turned off. So just to kind of run through this one more time. A repressible operon, where's my cursor? Here it is. A repressible operon, like the operon that we just talked about, is one that's usually on. The binding of the repressor to the operon shuts it off, and that only happens if you have plenty of the product already around because maybe your human ate plenty of tryptophan. So that's actually anabolic. So I have this ridiculous picture because sometimes people remember anabolic steroids are those synthetic steroids that some people take in order to get huge. So this huge person has built a lot of muscles, meaning they're making a lot of stuff. Anabolic just means you're making molecules. So the operon is anabolic, meaning you're starting with a precursor, you're using enzymes in order to produce something, in this case producing tryptophan. Inducible operons are usually catabolic. So catabolic, if you remember, means you break stuff down. So the picture I usually use for that is a cat that's eating something because cats are scary. And so it's breaking down or digesting molecules. So these are called inducible operons, meaning they're usually off, but you can turn them on. Because both of these use repressors, see here's the repressor, because they both use repressors, we're going to still call both of them negative gene regulation. So negative gene regulation just means that you have a repressor, whether that repressor is usually turned on or usually turned off. So let's talk a little bit more about the lac operon, which is the example of an inducible operon or a catabolic operon. So here's the lac operon. The way it works is that here's the regulating gene, and that's going to make a repressor. The repressor would bind to the operon just like before. Here are the enzymes LAC-Z, LAC-Y, LAC-A. LAC-Z codes for beta-galactosidase, which is the enzyme that breaks glucose from, I think it's galactose. It breaks down your disaccharide into a monosaccharide. Two other enzymes that you're going to need are permease and transacetylase. So just like the other operon, the mRNA is all transcribed all together, but the mRNA will have a start codon and a stop codon just like before so that you make your enzyme separate. So this polypeptide is made separate from the other polypeptides. But if you have this RNA around, you're going to be making them all. So the RNA is one continuous long piece of RNA, which is kind of weird, with these start and these stop codons so that the ribosome can bond here, translate the entire enzyme, and then fall off. Then you'll have another start and a stop, and you'll make the other enzyme that you need. The whole lac operon is the structural enzymes, or the structural genes, which are these, which need to be made all together or not at all. You'll also have the promoter, which is this part right? And the promoter is where RNA polymerase binds, and it also includes the operator. So by itself, the lac repressor is active. So here is the regulatory gene, which makes RNA. This, just like the last operon that we talked about, is always on. The regulatory gene is always transcribed at some low level, always translated at some low level. The difference is that this repressor is usually active. It's made default in the active position. So you're going to constantly have, normally have this repressor bonded to the operator. So this whole operon is generally in the off position. It's called an inducible operator because you can pull that repressor off. So if you happen to be in an intestine of somebody who just drank milk, that milk has lactose in it, and one form of lactose is allolactose. We're going to call lactose the inducer because if this allolactose binds to the repressor, which is always around a little bit, right, the repressor, if they're together, now you make this repressor inactive, which means you pull it off of the operator, and if it's off, now RNA polymerase can bind to the promoter, go ahead and transcribe these genes. Here's the mRNA of the operon, which has start and stop codons, so you can make this polypeptide, and then this part can be translated and you can make that polypeptide, and then this part can be translated and you make this polypeptide. So if you have lactose around, you're going to make all the enzymes you need to break it down. So that's called an inducible operon because it only gets turned on in the presence of the inducer, which in this case is lactose or allolactose. So here's a quick picture of what lactose or beta-galactosidase, beta-galactosidase. So this lactose is a disaccharide. It's going to fit into the active site of this enzyme. And once it's there, then they'll get broken down into their monosaccharides, glucose and galactose. So if the sugar lactose is around, it induces the bacterium to produce lactase and the other enzymes that it needs to break down all this sugar that's around. Otherwise, lactose isn't made in the first place, and it would be a waste of time and energy to produce it. Going back to the tryptophan one, repressible enzymes are usually part of anabolic pathways. So their synthesis is repressed if you have a lot of the end product around. So the cell needs tryptophan, but if your human is eating a lot of tryptophan, then you don't need it, right? So you're not going to produce. So high levels of tryptophan are going to turn off the operon, as opposed to the lactose one, high levels of lactose will turn it on. Okay. So summary is that operons that use these operons use negative gene control because they're switched on and off by a repressor. So it doesn't matter whether that repressor is normally made inactive, like the tryptophan one, or normally made active, like the lac one, like the lac operon. If you have a repressor, we're still going to call it negative gene regulation. Positive gene regulation is more like a volume control. So if you, like with lactose, for example, if you have lactose around, you're going to turn the operon on. But if lactose is your best food source, you're going to turn it on at really high levels. If there's something better to eat, you might make a little of the lactase enzymes, but not a whole lot. So this, not every operon is subject to positive gene regulation. Again, I want you to think of positive gene regulation as volume control. If lactose is your best food source, you're going to just amp it up and make a lot of the enzymes to break it down. So some operons are also subject to positive gene regulation or positive control through a stimulatory protein, such as CAP. CAP is short for catabolic activator protein. Please don't memorize this, but it activates transcription. So remember how contestant number 5 had this extra thing compared to contestant number 4? So this part of it, being able to turn the lac operon on or off, this was really negative gene regulation. This part, if glucose is around, you're not going to really use too much lactose. That's positive gene regulation. It's saying, okay, I could use a lot of lactase enzymes, but I have something better, so I think I'm going to turn the volume down on this whole lactase thing. So let's look at how that works. So normally, if you have lactose around, so let's say your human has eaten lactose right here, you're going to inactivate the repressor, which pulls it off of the operator, and so now RNA polymerase can bind, and you're going to go, go, go. Let's say that lactose is your best option. So you've got a lot of lactose around. Glucose is actually an easier thing to eat. It's just a monosaccharide. You don't even have to break it apart from a disaccharide to monosaccharide. Glucose is actually a better thing to eat. But let's say glucose is low. That means you want to make a lot of lactase. So what happens is you've got this CAP, this catabolic, what does it stand for again? Catabolite activator protein. So you've got a lot of this CAP around. It's normally inactive, but if you have cyclic AMP around, they're going to join together, and now you've got active CAP. So when glucose, which is a preferred food source, is scarce, meaning you don't have much glucose, lactose is your best shot. CAP is going to be activated by binding with cyclic AMP. Remember, cyclic AMP is a very common second messenger that we talked about several units back. So if you don't have much glucose present, if glucose is very scarce, you're actually going to have a lot of cyclic AMP around. So now CAP is binding to the promoter, and it's actually going to help RNA polymerase bind at a higher level. So activated CAP attaches to the promoter of the Lac operon and increases the affinity of RNA polymerase. Increases the affinity means it's much easier for RNA polymerase to bind to DNA. Affinity means how closely they attach and bind. So that's just going to accelerate transcription. So this is just a volume control. You're ramping it up. So already you had RNA polymerase able to bind to the promoter because the operon, because the operator was free. You pulled this repressor off. So you were able to transcribe and translate already. But now you're just transcribing and translating at a much higher level because RNA polymerase now has a higher affinity for the promoter. It's able to kind of hold on to it better because CAP helps it increase its stability. So now what happens if glucose levels increase? So the bacteria that use glucose instead of lactose in this case are going to be better off because it's easier to digest glucose than it is lactose. Lactose takes more steps. Any bacteria that can break down glucose when they're both present is going to be more fit than bacteria that break down lactose instead of glucose. So when glucose levels increase, you're not going to have cyclic AMP bonded anymore. So now CAP is going to be inactivated. It's going to come off of the promoter. And now RNA polymerase can still bind, but its affinity isn't so great anymore. So you'll still make lactase at a low level, but your volume is really turned down. So when glucose levels are high and lactose levels are high, you still don't have anything stopping you here. It's just that RNA polymerase doesn't bind quite so well. So you're still going to make some lactase, but not very much because glucose is so much easier to eat. So here's the summary of the Lac operon. If, let's say, you have no lactose around in either of these cases, whether glucose is around or not, if there's no lactose, then your repressor is going to be binding to the operator, and you're going to be doing no transcription of lactase and the enzymes that you need to break down lactose. So that makes sense. Let's say that lactose is present, though. If lactose is present, now your repressor is not going to be binding anymore. So you can go ahead and do transcription. So in both cases, you're going to be doing transcription of the Lac operon and making the enzyme to break down lactose. But let's say glucose is around. If glucose is around, that's really a much better food source. So it would make sense to turn down transcription of the Lac operon. And so CAP is not going to bind. And so this is just the volume control. You've turned down transcription of the enzymes that you need to break down lactose because you have glucose around. Glucose is a much better food source. So let's say you have no glucose around, but you do have lactose around. Now the CAP is going to bind to the promoter, help RNA polymerase to bind to the operon, and you're going to have lots of transcription of the lactose operon, which makes the enzymes that break down lactose. So negative gene regulation is the whole repressor idea. Positive gene regulation is the whole CAP idea right here. So CAP helps regulate other operons as well. So maybe there's not just a Lac operon, maybe there's a sucrose operon, and maybe there's a I don't know, name your other favorite disaccharides. So if you don't have lactose, you're going to be blocking the lac operon, but maybe you do have sucrose around. So if there's no glucose, CAP is going to be turning every possible other catabolic operon on because glucose isn't around. So in operons, bacterial genes are transcribed in groups. So one messenger RNA contains the coding sequences for several genes. So that's how they're either on or they're off. It's all or nothing, which makes sense because there's no reason to make LAC-Z gene, LAC-Z enzyme, so there's no reason to transcribe this one if you're not transcribing the others as well.